Dr. Alan Moy, and I'm the director and founder of the John Paul II Stem Cell Research Institute. And I'm going to talk about our genetic research program. To illustrate that program, I'm going to share with you a story about how our institute helped a family find a treatment for a rare, fatal neurological disease that's affecting 2 of their children. And in the process, we developed technology and a system that we feel can help other patients with genetic diseases and help patients with Alzheimer's disease. Our Institute's mission is to establish a faster and a more streamlined process to find cures and therapies exclusively using adult stem cells, develop technologies that will broadly advance drug discovery and regenerative medicine for many diseases, and increase the number of scientists and future medical practitioners that will work with adult stem cells with an emphasis on medical bioethics that value the life from the moment of conception to natural death. To illustrate our unique program, which is depicted in this slide, the Institute will conduct internal research and development and coordinate collaboration between the private sector, industry, academia, and government to advance research along the regulatory pathway to satisfy the FDA to ultimately find treatments and cures for patients. The Institute has identified 6 proposed research themes: regenerative medicine, drug discovery for clinical diseases that lack adequate treatments, personalized cancer therapy, the establishment of a public cord blood bank, the establishment of ethical fetal tissue bank for biomedical research, and the creation of new technologies for socially responsible biomanufacturing, and lastly, drug discovery for orphan rare genetic diseases. This is a story about Brian and Laura Hadley, who have 3 children, 2 of whom, Peyton and Kayla, have a condition known as Niemann Pick Type C, which is a rare fatal neurodegenerative disorder. In 2008, the Hadleys approached the Institute in search of a solution using adult stem cells. I would like to share with you their story and how the Institute contributed to finding a treatment for their children.
So when we first found out about the disease, we were finally confirmed diagnosed by a neurologist from OHSU. And he gave us the firm diagnosis of Niemann Pick Type C, which is a neurological degenerative disorder that is terminal, and we were told there is no cure.
So Brian and I met here in Medford. He came from Salem, Oregon, and I came from San Jose, California.
We worked together and started dating, and were married soon after that. Since that time, we've been blessed with 3 wonderful children.
Peyton, who's 12 years old, Kayla, who's 9, and Jonah, who's 7. Well, I noticed in 3rd or 4th grade that Peyton was having difficulty looking downward. So when he would type, he would cock his head, and that was one of the first indications. Prior to that, though, Kayla, as an infant, had an enlarged spleen, and they could never really figure out what was wrong. She was tested for many other rare diseases, and she happened not to be tested for Niemann Pick Type C.
Niemann Pick Type C is a cholesterol storage disease, and your body naturally produces it in the cells. So it's inside of the cells, and they get trapped inside of the cells because the NPC1 is a protein that transports that cholesterol out of the cell into the bloodstream. So that's what's wrong with the NPC patients, is that NPC1 protein is misfolded, so it can't do the job and take the cholesterol out of the cells.
And so it builds up in the spleen, liver, and the brain. Probably the most important part is the brain, because it creates neurological problems, including speech problems, swallowing difficulties, walking, talking, and eventually the children have to be in wheelchairs, and they considered to be what's called laid up, and basically, sitting all day long. We have to trust that he will either provide or he won't provide a cure, and there is a reason for that, and there's a master plan that he has. And maybe he is working through these children in a way that we don't understand.
So that's what we hope, and we pray for God's will and guidance for this. And if this is our plan, if this is what God's using our children for and us for, is to fund research and to help our children and all children with this disease, then we hope that that's the mission that we're given.
Cholesterol problem that me and my brother have. The cholesterol sticks to the cells, and the doctors are trying to help us, so if you can pray for us and pray for them, thank you.
This is a good example of how patients one day may be faced with the moral dilemma of choosing a cure that conflicts with their moral conscience. What impressed me most about Laura and Brian was their refusal to compromise on their strong moral belief in the sanctity of human life. Finding treatments for rare orphan diseases pose several challenges. There are over 7,000 rare orphan diseases defined as diseases in which there are less than 200,000 individuals affected by those conditions. 85% of rare orphan diseases are due to genetic mutations, and less than 1% of orphan diseases have a viable cure or treatment. There is very little funding, there are few scientists that work on these diseases, and it's very difficult to have viable animal models that are representative of these human diseases. There are challenges in finding enough patients to undergo a clinical trial, and there's a lack of commercial incentives for industry to develop a treatment or cure. Lastly, insurance companies frequently refuse to pay for these treatments even when they're available. The strategy of finding a treatment requires an FDA-approved drug. Before there's an FDA-approved drug, there's a need for drug candidates. Before there's drug candidates, there's a need for identifying a human drug target. And to obtain a human drug target, you need a targeted cell carrying that disease. To find a treatment, we first need to create a human cell model of that disease. In this particular case, a neurological model. We next screen a drug to identify one that works, test the safety in an animal, and then move directly to human testing. Unfortunately, there is a lack of disease-specific neurological human cell models. The largest repository lies in government, and they only have blood cells and skin cells, which are not representative of neurological diseases. And these cells do not have the growth potential for producing large number of cells for screening drugs. Stem cell models would be the best scientific approach to find a treatment. The usefulness of embryonic stem cells is a myth and fallacy because they do not contain the genetic characteristics of that disease. In contrast, adult stem cell would be the best model because they do contain the genetic characteristics of that disease. Neural stem cells from the brain would be too invasive to be used as a cell model. Thus, non-neural stem cells would be less invasive and be a more appropriate model for finding treatments. We and our partners have created fat-derived stem cells from routine liposuction. We have developed technologies that convert these stem cells into immature neurological cells. As shown in this movie, these stem cells convert within 1 day into neurological cells. When we convert these stem cells into neurological cells, there's an increased expression of an important enzyme called tyrosine hydroxylase that produces dopamine that is lost in patients with Parkinson's disease. We first had to solve some technical problems. Never before had fat-derived stem cells taken from a patient with a genetic disease been produced. As shown in the video, these children are very thin without much fat because they are on a low-fat diet. And we had to figure out how to keep these stem cells alive. The children had to have fat removed, so we referred them to Dr. Al Cram, a partnering plastic surgeon at Mercy Hospital of Iowa City.
So Alan had called me and asked me if I could help him get some cells from some children who had Niemann Pick disease. And we had some luck getting cells from other individuals by using a technique that's basically a liposuction technique. This was especially appealing for the children because we didn't really want to leave them with any physical scars from the experience. We then looked them over to see where they might have some fat, because we are going to get the adipose-derived stem cells from their fat. And their best spot really was on their thigh, the upper leg.
We were able to produce tens of millions of stem cells from each of these children. These cells were subsequently studied at the National Institute of Health and in academia. And they were found to have much higher cholesterol content than stem cells from normal volunteers. And we discovered there was already an FDA-approved drug called cyclodextrin that could lower the cholesterol content of these children's stem cells.
No, Dad's not even here yet.
Oh boy, he is.
So it's April 2011. We're at the Rogue Valley Medical Center, and our kids are next door right now having cyclodextrin infusions. We hit our first goal. We wanted to have a compound available by January 1st, 2011, and we now have that compound. It's called cyclodextrin.
Cyclodextrin is a sugar molecule, and the cholesterol fits right inside of the ring of the cyclodextrin compound, and it carries it outside of the body.
So Peyton and Kayla are the 5th and 6th child now to receive the cyclodextrin treatments, and the other kids are showing some good improvements.
Peyton has been on cyclodextrin for 3 months now, and we've seen that he's smiling more, and he's more affectionate, and he also engages in conversation with people, and other people have made those same comments.
Certainly there's been no additional neurologic decline, and there may be some early improvements in his tandem gait, and possibly in his eye, extraocular eye movements. Again, those are somewhat subtle, but and we're trying to keep from observer bias. We're pleased to see no additional decline and some definite improvements.
We're excited to be able to start the cyclodextrin treatments, and there is a lot of hope, but there's still a lot of work that needs to be done.
So scientific research needs to continue in order to figure out the optimal dose, the optimal delivery, and method in order to bring this to other NPC children worldwide.
We have developed technologies now that have the impact of accelerating drug discovery for a number of other rare genetic diseases. These cells in particular will be good cell models for the drug discovery of Alzheimer's disease for the following reasons. Niemann Pick Type C is the most common cause of juvenile dementia. The brain pathology of children with NPC is very similar to the brain pathology of adults with Alzheimer's. So we're looking to use these cells as a cell model for the drug discovery for Alzheimer's. Our objectives for the Orphan and Genetic Disease Research Program include the following: recruit patients, doctors, and hospitals to participate in our program to collect tissue samples to produce adult stem cell disease models, develop a drug screening program in partnership with industry, academia, and government, create a program that facilitates the FDA regulatory process, establish a network of sites for clinical trials, and finally, create an advocacy program to get insurance companies to reimburse for those treatments. The John Paul II Stem Cell Research Institute capital needs are to raise $10 million to initiate these research programs. The plans for that money include budgets for lease and lab build-out, capital equipment, and the hiring of scientific and clinical personnel. One of our objectives is to develop the world's largest repository of adult stem cells for rare orphan and genetic diseases for drug discovery purposes. This is what we need from you. We need financial donations, volunteers, and hospital partnerships. We have created a founders program under the website www.jp2sri.org, in which we need at least 40,000 donors in the United States to give at least $250. We've also created a national Give Cures campaign, which is listed at our website www.givecures.org. You can download flyers from these websites. We ask you to publish them in bulletins or newsletters of your schools or churches, and we ask your priest or minister or school for $1,000 towards this campaign. You can use this video to communicate your message. If they cannot afford the financial donation, we at least ask if they would publish our flyer. Please join our Facebook page, tell your friends, and indicate you like the Institute and direct others to our websites. Lastly, we ask doctors and patients with genetic diseases sign up on our clinical provider and patient registry listed at www.jp2sri.org. Lastly, there's real hope without having to compromise our ethics. I thank you for taking the time to listen to this video, and God bless you.